Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. M. Lois Tiffany Endowment, and by gifts from friends of the program.
It's a great pleasure to be able to talk about my work and try to convince you that physicists have a lot of say about brain function. My wife is a biologist, or actually a medical doctor, and she thinks I'm completely wrong. So I'm going to try to convince you that I'm not, and maybe her too. Anyhow, so if you attended another meetings or another Saturday Morning Physics this year, you notice that physics is rapidly changing. Physics, real life science, in terms of progression or changes of the fields or subfields that it's interested in. We had earlier this year Len Sander talking about brain tumors and how can you detect brain tumors and how can you model brain tumors. But even last week, Jennifer was talking about optics, imaging, and application probably of those to the living cells. So what I'm going to continue along those lines and tell you something of why physics is interested in brain function and how we can perceive brain function as a part of a physics problem. I want to give you a brief summary of what brain is. And what you will have to excuse me for, I'm going to go very quickly through some things and very superficially. I'm not going to give you too much details because many of those details would be for one semester course. So brain, yeah, it has 10 to 11 neurons. Neurons are the cells that we think are doing computation. So there's a lot of cells in the brain. There is another type or many types of cells. Those are the glial cells or astrocytes. And what happens, there's actually 50 times more of them. And until recently, we thought that they are completely not important for computation, so for information processing. But now we are actually not so sure of this. So you see, if we have to look at 10 to 11 neurons and then another group of cells, then it's really extremely complex systems. Every neuron is connected to 10,000 other neurons. Yeah? So there's, again, a lot of connectivity and a lot of network. Yeah? So our brain is a huge network, and that's exactly what I'm going to concentrate on during that talk. The number of synapses, so synapses are those connections, just more biological name, in the cortex is 10 to 14. So, I mean, you should realize from just those numbers, there's no way I'm ever going to be able to look at all cells at once and even figure out what is the connectivity into the brain. It's just too complex. So here's another statistics. Length of myelinated nerve fibers. So in our brain, we have about 100,000 miles or 150 to 180,000 kilometers of those connections. So think about it. It's relatively small, yeah? Anyhow, what happens is you can go many, many times around the Earth. And here's a kind of funny statistics. It's like a light bulb. Yeah, we are into energy saving. So actually, our brain, just to compute and to live, uses about 25 watts. So it's a decent bulb, just for computation. Yeah, because there are two things. You have to support your brain to make sure it's living, and the other thing is just some computation. So 15 watts is goes for supporting, and 10 watts goes for the computation. There is also bad news. Yeah? During this talk, every one of us is going to lose about 4,000 neurons. And I just have to say, please don't sue me. It's not because of me. It's not that I'm going to do something to you. It's just the rate in which we are losing neurons. Yeah? Initially, we thought that all those neurons are just not replaceable. That means they're just really going away and this number is dwindling. Now we know that in some specific parts of the brain, there are new neurons being born every day. Yeah? Those parts of the brain, for example, hippocampus or olfactory bulb, our sense of smell. We don't understand why those parts of the brain are the ones that need it. We don't understand why other parts of the brain do not need it. Yeah? So this is part of the many, many questions that we cannot answer. So first, I'm going to give you a rapid summary of what we know about the brain and what we don't know about the brain. And then, of course, what I'm going to try to convince you during the rest of my talk, that physics has something to say about the things that we don't know. And so this will be a kind of theoretical introduction. Then we are also doing experiments in my lab. So I'm going to tell you about the experiments we do to check some of those ideas. And then we are going to apply it to a real brain, to actually memory formation and learning, and to show you that some of those ideas seem to do pretty well, hold up in the real brain. So this is a picture of an anatomy of the brain. And this is just one picture, one projection of the brain. It's much more complicated than that, but just to show you how many things we know. Yeah? So the brain is covered with cortex. Yeah? This will be the association cortex. So this is what we think makes us think and makes us process information the way we do. But here, for example, you have a cerebellum. This generates motor patterns. Yeah? It makes us move in a really coherent way that we can walk, run, jump, and so on. Yeah? Here, for example, you have corpus callosum. This is just a freeway connecting two hemispheres. This is one hemisphere, there is another one. So the brain is just cut in half. Yeah? So this is just a lot of nerves connecting the two. And here, of course, the spinal cord. So this is what makes the information come from all our muscles and send down from our muscles and our sensory system, skin cells, and so on, just so the brain can control and get the information from other parts. Again, by no means this is everything. I just picked a few random things just to show you what we know. So now actually let's concentrate on the cortex. So here is the picture of the cortex. So this is this very thin layer of the neural tissue on the top of the brain. And you see what we know how to do is how to look at the function of this cortex. Which part of the cortex does what? So for example, motor cortex does exactly what we think it does. It generates the program which allows you to move. Yeah? Somatosensory cortex processes the information from our touch, feel, and other modalities. For example, here in occipital lobe, you have a visual cortex. So that processes information coming from the eye. I should tell you, this is the front of the brain, this is the back of the brain. So our eye would be somewhere here and sends information all the way to the back of the brain. And here you have primary visual cortex and secondary visual cortex. But again, it's extremely complicated. So for example, there are two streams in the visual cortex. One goes up and one goes down. So it's a really complicated system. And now we can go down the scale. We can actually look at smaller building blocks. So here are the neurons that I mentioned before. So this is a picture, or actually a drawing of a nerve cell. So you see, this is the cell body, and this is the axon. This is the wire I was talking about. And this wire goes to the next cell. So this would be a presynaptic neuron because we have a synapse here. This is the connection between the two neurons. This is the way that neurons communicate through the synapse. So what happens is neurons send action potentials. This is a picture of an action potential. So actually, it's just a depolarization, electrical depolarization. It's about 100 millivolts. So what happens if this neuron gets the information from other neurons and it gets depolarized, so the membrane potential grows, if it reaches some threshold, then about right here, the cell would generate an action potential, 100 millivolts, and this lasts about 2 milliseconds. The width of this is about between 1 and 2 milliseconds. Here you have actually a movie of what is happening. So the red is the depolarization. Red is the increase of the voltage. So let's start here. You'll see here becomes red first and then very quickly propagates down the axon, yeah, and it branches off. So here would be the postsynaptic neurons. They're getting all the information. Yeah, so again, here we generate action. Here we generate the action potential somewhere around here. It propagates very fast through axons and dendrites. And there are many, many, many types of neurons in the brain. Yeah, there are excitatory neurons, so those would be the neurons which actually excite the cells that are at the end. But there are also inhibitory neurons that actually inhibit the activity. So if it would be inhibitory neuron, this neuron would actually stop the activity of the next neuron. So going down the scale, so this is the synapse. This is the connection, yeah. And this is just this is like a chemical factory. The processes that are going on here are extremely complex and extremely hard to understand. Again, just to explain you in detail what the synapse does, it would take probably a semester of lectures. Yeah? And actually, to just tell you what we know, is probably more that we don't know. Yeah? So what happens? This is the neuron, the cell body would be here, so this action potential, this electrical action potential is being sent to the synapse. And then actually it's very funny because this information, this electrical information, is converted into chemical information. So something happens here that makes the synapse secrete neurotransmitter. This is a chemical. Yeah? You see those neurons are actually not touching. This is just a free space. So these chemicals go into the post-synaptic membrane. So this is the other neuron. This would be the other neuron here. And then they do something to this neuron to convert it, chemical information, back to electrical information. So we have electrical information coming here, converts to chemical information when it secretes the chemical, and then it's converted back to the electrical information. Extremely complex process. So as you see, we know a lot. We know how to measure. There's thousands of scientists working on it for a very long time, hundreds of years. Yeah? So what we don't know about the brain? Where starts to be a problem? So as I told you, neurons are the units that compute information. So the problem is, how do they do it? There are a lot of them. So it seems impossible that one of those neurons would really matter too much. Yeah? Especially if I told you that we are losing all those neurons per hour. Yeah? If one neuron would be that important, then actually we would be really in a bad situation. Yeah? Because after 1 hour, you would not be able to do things that you are able to do. So obviously, one neuron does not matter so much. So what happens is an ensemble of neurons that matter. So that means ensemble of neurons codes information, many of them. That's, again, exactly where I'm going with this talk. So the question is, how come, if I show you this picture, the association that I'm hoping you're forming is of that sort? Yeah? You have no problem very quickly recognizing what is on this picture and coming up with some decisions and functions, whether you want to buy it, not buy it, smell it, not smell it. Yeah? So this is the input. This is what your visual system is seeing. It goes into the brain, gets converted through some neural code, some way of spiking. So every line actually shows a spike. This is this action potential that I was talking about. So action potential or spike, I'm going to use those terms. I'm going to interchangeably. Yeah, so this is the spike or action potential, and just the ordering of those spikes and when they are being sent, we think is the code. Yeah? So again, that's the input, that's the I, it gets converted to that, and then the brain has to make some decisions. It's being converted again to other program, against spikes, and which makes you smell. But you have to do it extremely rapidly, yeah? Because if I change the input on you, yeah, well, you wouldn't want to do that right now, yeah? It's maybe the last smell you take in your life, yeah? But you did it very quickly. Yeah? You start laughing almost immediately, yeah? So the better program for doing this is this, yeah? Well, but what's the difference? What's the difference, yeah? Well, you know what is a lion, but it's a very short process that you're going through, yeah? Because from point of view of our retina, of what happens in our eye, this is just a set of lines and colors, nothing more. This is just a set of lines and colors, yeah? So that means all these neurons that they do then convert this information into something that's meaningful for you. This is a set of your experiences, yeah? If I would, from day one that you're born, tell you, lions are good, they're like kittens, you can smell them, yeah? You could kind of, when I would show you these pictures, you could think, okay, I can smell the lion, yeah? And if I would teach you from the start that, no, those are really nasty flowers, they eat people, yeah? Well, you'll be running away from this flower. So you see how complex is the processing you're doing, yeah? It's not just your visual information, it's all your memories, all your experiences go through your life, go into interpreting this picture to generate an action. So how do we do that? The first question is, we don't know, yeah? But that will be the end of the talk, so I have to tell you something more, yeah? So what we think is happening, there are two basic ways we think we can compute information. And the answer is that probably none of them is exactly true, and both of them are applied. But I'm going just to give you the two opposites. So let's look at the hierarchical coding. So the idea here is that our brain is a layered device. So you have neurons on different layers. You see one layer, second layer, third layer. Those layers are interconnected, but they're not so much connected between them on the same layer. You see, so those connections are being just sent to other layer. So what happens here is that the most upper layer processes the easiest information, the most simple information. So for example, those neurons would process color, whereas those neurons would process shape. And then you go to another layer, they send all those connections and the information back. So now this layer computes a more complex information. It produces, so this is a rectangle, this is red, so this would, for example, compute red rectangles, yeah? And so on and so forth, yeah? So this layer could compute red rectangles that are moving, for example, yeah? And so on and so forth. That would mean that at the end, and you know you have convergence, you have many more cells here, less cells here, even less cells here, that at the end you would have this one cell or very small set of cells that would code for particular object, yeah? So for example, I'm looking at you right now, yeah? So if this would be true, I just right now formed a set of cells that are coding you and only you, no one else, yeah? But then the basic problem with this idea are two basic problems. First of all, that's really a hardwired computation, yeah? I have all those connections, they are really hardwired, so it's not easy to change the program, yeah? You have to rewire everything. The second, maybe even bigger problem is that I just mentioned you, that I just form your representation, those are the cells that are coding for you. But there are 250 of you here, yeah? If I'm going to form representation for every one of you, and then let's say everything that happened yesterday and everything that happens tomorrow and so on, I'm going to run out of space. Yeah, so I would have to carry suitcases with additional neurons that would store all this information. So that's not a good idea. We don't do that, and still we remember things. Yeah, so that's good for us. Yeah, so we think there is another way we can compute the information. Yeah, those would be. We start the same way, but you see that the network here is not so well organized as the network on this side. Yeah? So the circles are neurons, the lines are the connections. And as here you saw very good organization of what is happening, here you don't see that organization. Yeah? You just see a mess. So let's assume that actually those cells are coding for the color and those cells are coding for the shape. So what's going to happen now, if you want to put those two together, it's a completely different process. You make them somehow talk to each other, yeah? And then the other cells recognize that there's something in common and they code for the red rectangle. So the interaction of those two cells, of those two groups of cells, make the next layer, the more complex information. What is the big advantage for this? First of all, this is really a software type of processing, yeah? So there is some software that allows us to connect those two groups. So it's much easier to change it. Yeah? I can just rewrite the software. Yeah? It's much easier to rewrite the software for the computer than to build a new computer. Yeah? And also the other thing, let's say I will introduce another color, green color. And now I would like to process an information of a green rectangle. I don't have to code for another rectangle. I just can make those two groups come together. So you see I'm saving space because the cells that actually computed the rectangle are being reused to compute a green rectangle as they were used before to compute the red rectangle. Yeah? So that actually significantly reduces the need of having the suitcase. Yeah? So let's try to see how does this work. What do you see? So some of you are going to see an old woman. This will be an eye of the old woman. This will be the mouth of the old woman. This is the nose with the wart of the older woman. This is the hair. Yeah? That will be the first picture. Some other people are going to see a young woman. So this will be a neck of the young woman. This is the nose of the young woman. This is the ear of the young woman. And she's kind of looking backwards. Yeah? So she's looking away from us. Everyone see both of them? Yeah? And this is the necklace. But notice what happened. I did not change anything in this picture. The picture is as it was from the beginning, yeah? So that means you can interpret the same features on the same picture in completely two different ways, yeah? Once this is an eye, other time this is an ear. Once this is a nose, once this is a wart. Big difference, yeah? Once this is a mouth, once this is a necklace, yeah? You have no problem. So that means you take this piece and assign it completely different meaning to see this picture. So that seems like a software, yeah? You do it extremely fast. You can now switch, if you see both, you can switch as fast as you want. Old, young, old, young, old, young. Happens very fast. We cannot rewire our brain in that time really significantly, okay? So that would point to a software processing. Yeah, so this is just a summary of what I just told you. Yeah, same picture can be interpreted in two different ways, but you can see one at the same time, but you can switch very rapidly. Okay, let's look at another example. What do you see right now? That's less tricky. You see, it's obvious what we see. Yeah? So what you should see, or I'm hoping you see, is that you see a white triangle covering also a white triangle with the black borders. You agree with me? That's what we see, kind of? Okay. Well, would you agree with me now? Yeah? Kind of this white triangle is gone. So what did you do? What did you do on this picture before? You added the information that was not there actually, yeah? I just took out those black circles, yeah? So what your brain did, said, that makes sense. This has to be a triangle. But this information was never there, so we tricked our brains of thinking this is the rectangle. This is a triangle, sorry. Yeah? So what happens is that even if some parts of information are missing, but the brain thinks it should be there, it's going to add it for you. Yeah? Hey, you're not seeing everything. I will give you all the rest. Yeah? Most of the times the brain is right. But sometimes, obviously, the brain is wrong. Yeah? So that's exactly what it says. Yeah? And it turns out that this model of information processing, at least in those examples that I gave you, makes more sense. So again, we are activating two different parts, yeah, that code for different aspects of the same figure, and then we are, through software, combining them to form one. There is actually one problem with it. How do we do it? That's what's interesting. How the brain knows that those two things talk together to form a red rectangle. How? What's so specific about those cells? Because sometimes they're going to talk to each other, but some other times they're not. So what makes the brain quickly decide they're talking? This is so-called feature binding. Yeah? We are binding features. We are binding the shape with a color. Those are two completely different independent features. We have to put them together to know that this is the red rectangle. How does this process go? So that's actually what I'm going to try to tell you. So now let's change actually gears a bit. And now I'm going to tell you how I view as a physicist brain. Yeah? So everything that I told you right now is actually a biological problem or a psychological problem. Yeah? Or cognitive problem. Now let's try to put it in the language that I understand better. Yeah? So the physicist view of the brain. So let's summarize what we know. Neurons fire action potentials or spikes, or these very brief events. The other thing, neurons are to some degree unique. I told you we have excitatory neurons, we have inhibitory neurons. They are coupled to many, many other neurons. Many, too many for our purpose. Yeah? So what we can say, the neural network is weakly coupled. They are weakly coupled similar dynamical units. Yeah? So we have a network of them. Yeah? So we have a lot of neurons that interact. And that already brings it on the level that I understand better in terms of physics of it. Because it has been studied for a long time. So first, let's answer the question, if we have only two of them, coupled similar units, what are they going to do? And the answer is, they're going to synchronize. Yeah? So what happens is, if we start with two oscillators, actually I'm going to show you a demo right now. So physicists think that neurons could be oscillators. Yeah? So here is an example of two oscillators. So what you see, here is one oscillator. I can make it move, go up and down. And here is another oscillator. Right now they are decoupled. There is wire that's missing. This wire has to go here. Yeah? So you see, this one is moving. This one is not doing anything. But we have here, this thing is a coil. And at the end of this oscillator, magnet is hanging. Okay? When the magnet goes inside the coil, the magnetic field changes. This coil generates a current. This current is going, when I connect this wire, it's going to be sent to the other end. And then we are going to generate current in this coil. When we generate current in this coil, we change magnetic field. Yeah? So now we pass on the information about what this magnet is doing to this magnet. Look what happens. Again, look right now. Nothing is happening because the cable is disconnected. Okay, I'm going to stop that. I'm going to plug in this cable, and we're going to try again. Look, the other one starts moving. Not only it starts moving, it's synchronous. They're doing the same thing. That's what synchronous means. They're doing the same things in time. They're going both up and both down at the same time. This is synchronization. And actually, believe it or not, I would argue it's not such a bad model of a neuron, as ridiculous as it sounds. Yeah? Because what happens, I have neurons that fire action potentials all the time by themselves. So we can say the movement up and down when the magnet goes into the coil is when the neuron would fire action potential. Then this is the time when it sends the information to the other end. This is the time when this neuron knows about it and starts doing the same thing. I can trick those two oscillators. What I'm going to do, I'm going to switch cables on them. So I'm going to connect now the black cable down and the red cable up. I'm going to stop both of them, and we'll try the same experiment again. Look what they are doing now. They are anti-synchronized, anti-phase. This goes up, but this goes down. This goes up. It's also a type of synchronization, just a different type of synchronization. Okay? So you see this is very, very common in nature, that the couple elements synchronize. Very simple experiments. Yeah? So here is the trajectory. Yeah? This is going up and down, oscillator. You see, in this example here, they would start at different, not synchronized, they would do different things. That's why the green line and the blue line do not overlap. And with time, they would start doing the same thing. You don't see blue line because the green is just on top of the blue. That's why we don't see it. Yeah? This is another example. Slower oscillator. Yeah? Here is actually example of neural example. It's a computational model of neurons, but it's a pretty good computational model of neurons. Yeah? So again, they fire spikes at different times, but then the timing of those spikes synchronizes, so they do the same things. Again, you don't see blue because the green is on top of the blue. It's just not there. I mean, it's there, but you don't see it. Okay, so it seems we can grasp what the two are doing. But there's a big problem, and I'm going to show you. What happens when you have a lot of them? So what you will see, what happens when you have a lot of them actually is that they can be connected in different ways. The structure of the network can change. Because, for example, in this crowd, yeah, one person can talk only to another person. So this is a nearest neighbor connection. You can talk only to one person to your left or to your right. So if I would whisper something on your ear, and I wanted person in the back to hear, it would have to go all around the room until it reaches the back. Yeah? Okay, it would take some time, and actually I could see the dynamics of the information processing through the room. Yeah? But there's another option. For example, you have random connections. You can take your cell phone and call a person in the middle of the room, or call a person on the left, or call a person on the right. Yeah? Then this information spreads very, very fast. Whole room, very soon, without this progressive information transfer, would know about it. And this is exactly what happens in the brain. In sense, you can have only local connections. So you see nearest neighbors, those are the neurons, this is two-dimensional network. Only the nearest neighbors are connected. Or, on the other end, you have completely random connections. Any neuron connects to any four other neurons in the network. We don't have a control over it. And then there are all the possibilities in between. In some range of those possibilities, we call those types of networks small world networks. Those are the networks that actually have many short-range connections, because nearest neighbors connections, and some random. So what I'm going to do, I'm going to play you a movie of a network. Those are 200 by 200 neurons. So here are neurons and here are neurons. And what you're going to see color, when it's black, nothing is happening. You will see a color when they fire an action potential or when you have a depolarization. And you're going to see it for completely local connections, for some mixture of local and global and random connections, and only for random connections. Let's start with the local connections. Okay? So you see, this is when we stimulate. This line that you see here, we stimulated a group of neurons. And then the information spreads. Yeah? Those neurons cannot fire too closely action potentials, and we have periodic boundary conditions. So when it disappears here, it appears on the other side. Yeah? And what you saw, there was one place that we activated. The information propagated as a wave, came from the other side, and then disappeared. That's it. You will see it in a moment again, it will just replay. The movie is going to replay. Let's look at the mixture. Yeah? So now, in this picture, what I'm doing, I'm just putting some number of these random connections, very small amount, 10%. Again, I stimulate. The same simulation, nothing changed. I stimulate. Now look what happens to the network. First of all, completely differently. That's what I told you with the example of this crowd. The information would spread very fast. Yeah? We wouldn't see this propagating wave. But there's another interesting thing. Not only it spreads very fast throughout the network, it never stops. You see, that went away. That doesn't go away. We don't stimulate anymore. That doesn't go away. It's persistent. Yeah? It will go like this forever. Okay? Let's try the other example. Again, we stimulate. Yeah? You see, those will not go away too, but the dynamics of those is completely different than the dynamics of those. Yeah? This is kind of unorganized type of dynamics that just goes on and off at many different places. This is very well organized. This is synchronized activity. Yeah? They all come up, all come down. Yeah? So this was as the model of two. Yeah? So this kind of behaves like this example when they were synchronized. Yeah? This is different, and this is completely different. Believe it or not, we think that all of them happen in the brain at different times. So let's look at it. So now I'm going to tell you what we like to analyze in the brain function. So this is a neuron. The circle represents a neuron. This represents timing of action potentials. Every line is an action potential, or spike. Spike and action potential is the same. So I'm really interested in understanding the networks. Yeah? So now I have a position of a neuron in the brain, and I have information what it does, the action potentials. You see, this neuron and this neuron and this neuron and these neurons do almost the same thing. They fire action potentials at the same times. Whereas this neuron, for example, this neuron and this neuron, do completely different things. There is really nothing in common between those patterns. So then what I can say, okay, so let's look at more closely at those neurons that do the same thing. So those are the ones that became red. You see again, this pattern is very similar to this pattern, to this pattern, and to this pattern. And that's exactly what we think future binding is. So what happens, the brain has ways of detecting which neurons do similar things. And when the brain sees that some group of neurons does similar things in a time, or synchronizes, or this is very similar, it will say, aha, they code the same information. So let's say this is the red, this is the rectangle. Yeah? Grouping all of those together will mean red rectangle. So spatiotemporal patterning is the thing that binds the information together. The fact that they are doing the same things makes the brain think that they code for the same information. But as I told you, the things are extremely complicated. So now, because I cannot really know what all the neurons in the brain are doing, it's 10, as I told you, it's 10 to 11 to them. I cannot, there's no bloody way I'm ever going to figure out what are the connections between them. It's 10,000 per neuron, yeah? Our brain is really dense of those neurons and connections. There's no way I can record it, know it, which is connected to what. So in a sense, if I don't know it and I cannot know it, I'm not interested in it, because I mean, what can I do? Yeah? So in essence, what I'm trying to do, I cannot figure out the anatomical structure of the network that's doing something, but I can try to figure out functional dynamical structure. How does this work? What we are going to try to do. So we are going to group the neurons that do the same thing. By the same, we mean they have very similar patterns of activity. Again, those are spikes. This is time. Yeah? You see, so I group pretty well. Yeah? Those neurons do similar things. Those neurons do obviously different things. So those are the red neurons that do the same thing. So what I'm going to come up with some set of measures, mathematical measures, that's going to group them in a way that those 4 neurons will be together, and those neurons that do different things will be far apart. Yeah? This is the functional coupling. So just to make sure that we understand, by no means I am saying that those neurons that do the same thing travel in the brain. They don't. They stay in the same place. I am changing the question that I'm raising. I'm not interested which geographically neuron does something the same in the brain. I'm just asking the questions, what are the neurons that do the same thing? And then I can group together, and that forms a cluster. So now this cluster is the feature binding. Okay? So if I can figure out those neurons form this cluster, I will know which neurons code for the same information. Again, red rectangle. Okay? So now we kind of have to fill out the last part, go from feature binding back to the brain. But before we do that, you should say, okay, you have very nice ideas and so on. I mean, you have to prove that they're right in some way, I mean, before doing it. So actually, we do experiments. This is our setup. This is our setup in the lab. We grow our own networks. So what it means, we take neurons, put them on a glass cover slip. This glass cover slip has electrodes. Those are the electrodes in them. And then those neurons regrow connections. So those neurons, the cells they take, they regrow connections forming a network, random network. This network in terms of connectivity properties has nothing to do with this what is in the brain. That's actually a weak part, a minus of the situation. Yeah? But then those electrodes can record from different parts of the network. Yeah? So I can monitor the spatiotemporal pattern that I was interested in. I know what those neurons do. Now let's talk about size. You see, this is 1 millimeter. To give you a better impression of the size, I'm going to start zooming in. You see this black dot? This is where we record from. It's small. So you see, those white points are the cells. Let's zoom in on them. Yeah, this is a magnification. So now those are the 2 electrodes. You see different neurons and a lot of lines here between the neurons. So those are the connections. Yeah? Those big lines that you see, those are those wires connecting the electrodes so we can record anything. Yeah? So you see electrodes, wires connecting electrodes. Those thin wires are the ones connecting the neurons. Yeah? You see, look at this network. Even though I went out of my way to make this network much simpler to what is in the brain, it's still extremely complicated. Yeah? I cannot figure out exactly the way those connections work. Yeah? It's just too dense. I can zoom even farther in, and you see bigger neurons, even more nasty pattern of connections. But anyhow, it's easier. Why is it easier? Because I can record from those electrodes, so I know what different sites in the network do. So this is electrical type of recording, and I can also do optical type of recording. Okay? So this is the electrical recording. This is the movie of the activity of all the cells at those sites. So you see the pattern is exactly the same as the pattern of electrodes. This is just some kind of dirt. Yeah? And you see those are the spikes. So this is the actual recording from our cells. Yeah? And this magnification of one of those squares. Those are the spikes. That's how the spikes look in real life. So actually you see a pattern forming. They actually do a lot of things synchronized. Yeah? This is the example of the optical recording. So there are special labels, we call them dyes, that the cells can take them up. They go inside the cells and they stick to different things and report, for example, the concentration of those things. So this is calcium dye. So what happens, this dye goes inside the cell and reports the concentration of a calcium inside the cell as a function of time. What it does, how it reports it, it fluoresces light. Yeah? And what I can monitor the changes in fluorescence. When the concentration goes up, the fluorescence will go up. When the concentration goes down, the fluorescence goes down. I will not elaborate on that, but actually calcium is a very important ion in the brain function. It does thousands of things. One of the things it does, it slowly depolarizes the cell. So I know, so those are the traces from recordings from this culture. The colors are matched here and here. And you see the dynamics, so this is time, dynamics of calcium is completely different as a function of time in different sites, on different cells. Yeah? We know that the action potentials, the spikes, will be at the beginning on top, here and here and here and here and here. So we have at least means to look what this network is doing. Yeah? This is the dynamic, this is the temporal pattern. But we have to know something about the space. So we can label. We can actually attach the labels to different proteins that are expressed in different parts of neurons or astrocytes or synapses. So this actually labels for all the connections. You see, this is our MEA, multi-electrode array. Those are the electrodes. And this is just every dot, which are many of them, believe me. There are millions of them here, that actually stands for this connection, for the synapse. So you see, the only thing I can tell from here is not which one is connected with which, is there are different densities. There are denser parts of the network and less dense parts of the network in terms of connectivity. The picture here actually is a staining of a single neuron, or a couple of neurons. So this is the cell body, and you see the connections. So we can do both. We can stain globally the network, the whole network, or we can stain single cell or a couple of single cells and try to figure out something about anatomical patterning, the space about which we are talking. Okay, so now let's actually try to apply all this information to the brain. First of all, what we would like to do, we would like to measure whether those our ideas make some sense in real brain function, whether we can actually record them and to show that actually there are some changes that make sense in terms of our theory, or actually the vice versa, that our theory makes sense for the changes that we see. So we are going to look at learning and memory. What is memory? Yeah? Storing representation, that's a very dry definition that I came up with. But I can ask you a question. Where is your car? All of you that came here by car, you can picture your car, where your car is parked. Not only can you picture where is it parked, you can probably picture your car at the same time. Yeah? So I evoke a very complex pattern of your memories. Those who came here by bus or walking, they say, okay, my car is standing at home, or I came by bus, so irrelevant question. So what are the properties of the memory? It's fast. You park a car 1 hour ago, yeah, or just above 1 hour ago, yet you remember it very well. So it's very fast in forming. I mean, if it would take 2 hours to form a memory, you would not have no idea where your car is parked right now. You would be in trouble. Yeah? And you know it can actually be much faster. You go, let's say, to a drugstore or a grocery store, you go for 2 minutes, you exactly know where to come back. So it's very fast how it forms those memories. Yeah? It allows to retrieve a lot of ideas based on fragments. I ask you, where is your car? I bet many of you already had a picture of your car, color, make, whatever, type of car. Yeah? I just ask you, where is your car? You didn't just picture the parking space. You picture the whole complex information. Yeah? And it seems to never be full. At least that's what we hope. Yeah? We do forget things, but this is due to other things. Yeah? So what is learning in terms of network dynamics? So what we think is, and this is actually very old work, this very famous sentence, neurons that fire together, wire together, it was expressed by Donald Hebb in 1949. So you see, we already are working on this for an extremely long time. And what it means that the neurons that represent the same information make new connections, those are the red lines, or make the existing connections stronger. Yes, so I'm forming new connections or making them stronger. The other connections that are irrelevant are made weaker, or at least they don't change. Yeah? So on the level of the network, this is how the network changes to be able to store the information. Obviously, that's still very fast. Yeah? So now, this is what I just showed you before was the anatomical space, the real network. Yeah? But now, as I told you, I cannot measure real network in the brain. I have to talk about the functional network, yeah? The functional space. Remember I told you about the functional space. So what happens during memory formation in this functional space? This is what I'm going to try to detect in the real brain. Yeah? So those are the neurons that initially code for the same information. Read again. Those are the neurons that are irrelevant. So when you form a memory, you make in this functional space, again, this is functional space, make them come closer together to form a cluster, and those other ones are being pushed away. You see? So this is like improving a signal-to-noise ratio. If you bring them closer, it will be it's much easier for you to see this cluster, but you want to push those away to make sure that your cluster is not obscured. Okay? That's what we think the brain does. So let's try to measure it. We actually came up with a pretty complex ways of measuring this functional space. Yeah? This is the data from our collaborator, Josh Berg, in psychology. What he does, he implants electrodes in the rat's brain, and what happens, the rat can do many different things. It can walk in the new environment, in a new maze, or it can walk in a familiar maze, the maze that it saw before. Yeah? So for example, if I would bring this rat first time to this room, it will be a completely novel environment. Yeah? So it will start trying to figure out what's going on here, yeah? Trying to find something that he can remember it by. Yeah? But if I would start bringing it every day for 5-10 minutes, after third time, it would actually know this environment pretty well and say, this corner looks nice, I'm going just to go there and sit there because that's what I like. Yeah? And forget about the rest. I know the room. It's not interesting. I'll just sit in a corner. Yeah? So that's what he does, more or less. Of course, he doesn't bring them here. He has it much, much smaller, but that's the basic idea. So here is the example of the recording from his data. Yeah? So we have 80 neurons, and the rat is put into novel environment, and then it falls asleep in this environment, and then wakes up. Then Josh here puts it into the familiar environment, the one that the rat spends a lot of time in. Yeah? So we have those recordings. And then he can put it back into this same environment that was novel, but will not be novel anymore because it learned. Yeah? Okay, so now let's try to apply our analysis to see what happens. Now I have to explain to you what you see. So let's remember these networks. What I'm really interested in is the distances in this cluster between the red cells and the distances between the red cells here. And I'm interested between the distances between all the orange cells here and between the orange cells here. As I told you, the cells that code for familiar information will cluster together, so the distance will go down. So that means in the novel environment, this distance should be larger than in the familiar environment. And that's what we get. This is experimental result. This is a result from the recording. So this is not a theory anymore. That's the result of the experiment. You see, initially the distance between those cells was greater than the distance between those cells. Now so those are the red cells. Now let's look at the orange cells, those two bars. So first of all, the distance was much bigger between the orange cells here. But then I told you this works like a signal-to-noise thing, so we actually want even more to increase this functional distance, again, functional distance, yeah, between those cells. And that's what happens. Yeah? Now the blue is higher than the red one. This is what happens in the novel environment. That's what happens when the environment is already known, the same environment known. So again, this clustered, those neurons were pushed away. Hey, so that's what we expect. That's what we got. Very nice, yeah? It seems that all those ideas make some sense. So now what we want to do as a physicist to come up with a very simple model of understanding how this can work. Yeah? So how does our brain process the information? Before I go there, I have to give you some details about the biology. Because when you do modeling, you cannot just do any model. You have to do model that makes some sense in terms of biology that you know, that you're working with. Yeah? We cannot, actually, I really cannot publish an article saying that neurons are those type of oscillators. Yeah? People would laugh at me. Yeah? I have to put some real facts into a model. So it turns out that our memory, as you could imagine, is an extremely complex thing. And there are two more, but I'm going to concentrate on two basic parts that constitute our memory. One is the hippocampus. That's the picture and the position of the hippocampus in the brain. It's not blue, it's just marked blue. And here is the cortex. Hippocampus is responsible for storing short-term memory. So the fact that you remember where is your car parked right now is mostly hippocampus. Yeah? Hippocampus does that. Your cortex has right now no idea. Yeah? So what it does, it rapidly learns. You know where is your car. Yeah? And what happens is, you can think in a very, very far kind of analogy that the cortex is a stupid brother of the hippocampus. In some sense. In the sense that it learns much, much slower. So what the hippocampus has to do, it has to replay this memory on and on and on and on to the cortex. At some point, cortex will get it. Yeah? But cortex is much better in the other thing. Once the cortex gets it, it remembers it. Okay? It will never let it go. Not never, but let's say almost never. Yeah? And the cortex now is much smarter brother of hippocampus because actually it can process this information. So all the higher order thinking that it happens in your cortex. So in this sense, now hippocampus is a stupider brother of the cortex. Yeah? Because more advanced processing does happen in the cortex. So we're going to make a model of this. Yeah? Just before I tell you the model, what are the questions we are trying to answer? How does the hippocampus know when to learn a new memory? Because you can say, let's say you see me for the first time, so you want your hippocampus to learn my representation. But let's say you see me again in a week. Yeah? You already know me. There is no need for hippocampus to store this new information because your cortex already knows it. 1 week is enough to form representation in the cortex. So something has to tell hippocampus, hey, forget it, I know it, not interesting, do something else. Yeah? Okay. That's a very tough question. Yeah? The second thing is, how the hippocampus controls the replay? The same type of question. Okay, you learn the representation very fast, you start replaying it to cortex. Well, cortex is listening and listening and listening, and cortex gets bored. It already knows that they have to repeat it. That means it's over and over and over and over. It's like, okay, enough. Yeah? Well, what this enough means, what is this enough? How the cortex tells hippocampus enough. Yeah? And then the last question, okay, so now it's enough. So hippocampus can forget this memory. How come it does it? How it forgets this memory? Yeah? Again, not a trivial question. It seems trivial to us. It's clear it has to do it. We don't know how. Well, we did a slightly simplified model. What we have, we know hippocampus have both excitatory neurons and inhibitory neurons. The excitatory neurons excite the cortex, but the excitatory neurons in the cortex inhibit or excite inhibitory neurons in the hippocampus, but that means they inhibit, because the inhibitory neurons will inhibit the activity of neurons in the hippocampus, excitatory neurons in hippocampus. Yeah? It's really easier than it seems. So what we have here, this is time. On the x-axis is time. So I'm running my simulation. My simulation has 500 excitatory neurons, and let's say the same number of inhibitory neurons. You're only looking at the excitatory neurons because those are the neurons that actually send the information, as you saw in the model, to the cortex. Okay? Every dot represents an action potential. So now it's not a line, it's a dot. Okay? So now, what I want to show that when I form new connections in a network, the network is going to behave completely differently. So here there are no new connections. There are no red connections here. And I stimulate a group of neurons here, and then some other place, and back to the same place. This is the response of the network. So the neurons that I stimulate are active, not because I just stimulated them. But there's no difference between those two different places. Now, I add red connections in this part of the network, and I stimulate again. Now you see that this part of the network with the new connections actually responds completely differently than this part which has no new connections. Yeah? So hey, that could be the novelty detection. Yeah? If I form new connections locally, and then the network, this part of the network where I form those new connections, responds in so completely different way than it did before. This is the same, yeah, just new connections, and differently than any other part of the network. This is a good way for the brain to figure out, okay, much more activity, you know that, yeah? It's already familiar, you don't need to do anything more, yeah? Moreover, in some special circumstance, even if I don't stimulate, I don't drive with some neurons, What happens here is that can activate by itself. This memory, because there are additional connections, this memory can just activate to yourself. You know this, yeah? Let's say you eat breakfast, you start thinking about something, yeah? Comes from nowhere. No one asked you about it, yeah? This would be it, yeah? But actually what happens in real life during sleep, this happens a lot during sleep. That's why actually sleep, one of the reasons why sleep is so important. And maybe you noticed that if you read something just before you go to sleep, after you wake up, you remember it much better. That's why. Okay? But this is only hippocampus or this is only cortex. They're very similar networks. The only difference between the cortex and hippocampus right now is that one learns faster, one learns weaker. So now let's connect those two together. Let's make a whole network. So we have hippocampus connected to the cortex. Yeah? So now, if there is a novel memory coming on, hippocampus gets really activated, it sends information to cortex, but cortex doesn't know it, doesn't have those additional connections. So it kind of gets slightly more activated, but not that much. So, okay, that's it. Yeah? Now, hippocampus, when you have a memory stored in the cortex, hippocampus gets activated the same way, but now the cortex gets activated much more. But you have a negative feedback from the cortex back to hippocampus, so that actually stops the hippocampus from being as active as it was before. This is the way the cortex tells the hippocampus, don't worry about it. Go someplace else. Do something else. Yeah? Yeah? Here is the replay. This is this thing I told you when you go to sleep, you remember much better. Yeah? So you have 3 memories. Those are 2 novel memories. The blue and the red are novel memories. The green is more and more familiar memory in your cortex. What happens is, hippocampus can easily replay. This is the replay, the black, when they are really active, those neurons. Yeah? This is the replay of the memory. This is the familiar. This is the blue memory. This is, let's say, red memory. This is the blue memory. Because if the memory is familiar, cortex inhibits the replay. Again, just presses stop on the record player. Yeah? So it has the same, I didn't change anything in the network, it's the same network. Yeah? So I can explain a lot by interaction of those 2 networks and by the fact that I locally add new connections. It actually does 2 things. Yeah? I have a novelty detection, telling the hippocampus not interesting, don't bother with it, do something else. And I have a mechanism to stop press the stop button on the record player. I can put all those 3 together, and now I can look at the dynamics of the hippocampus and cortex together in a whole process. So more or less what I'm showing you here is that I can mimic the interaction between hippocampus and cortex during the whole process. When you see something novel, when you learn something novel, when you replay something novel to cortex, and then when cortex tells you do not replay it anymore and forget it. Yeah? Hippocampus can forget it. So in this extremely simple model, I mean, biologists would come and just yell at me that this is just an extremely simple model, just pinpointing one possible mechanism. I can learn very fast new memory, replay it to the cortex, make the cortex learn, make the cortex tell the hippocampus, you don't need to do anything more about it, and make the hippocampus forget everything by manipulating one parameter. Yeah? So we started from the brain function. I showed you some pictures that told us something about how the brain works. From this, we figure out what is the neural coding, what are we looking in this pattern of activity. Then we actually talk about network dynamics. We have experimental way of looking at this network dynamics. Yeah? And then we figure out what is the feature binding. And then we applied it to what we think is a real brain. Yeah? So we closed the loop. And that's what I wanted to tell you today. Thank you.
Saturday Morning Physics is sponsored by the Physics Department of the University of Michigan, by the Dr. Remlois Tiffany Endowment, and by gifts from friends of the program.